Hello dear friends, myself Professor Rishikesh and you are watching BSc Botany Studies Classes channel. Here in this video we are going to discuss about the continuation of our discussion. We will discuss first about the introduction of the molecular biology. What is this molecular biology? First of all, the science is originated in 1945. You should know this. The term molecular biology is given to this science because in this science, molecular biology I am talking, we study mainly about the molecules. The molecules are of two types, primary molecules and secondary molecules. But molecules are not called as molecules in biochemistry. They are known as metabolites. It is alternative name of molecules. The study of molecule, or you can say study of metabolite, is known as metabolome. You should understand this terminology. You should get familiar with these terminologies because after FYBC, SYBC, TYBC, you will have to study about MSc, and post graduation after completion you will have to face competitive exams. So these words will be there. So we are going to study about metabolites. The metabolome means study of these metabolites. So I have said there are two types of metabolites, primary metabolites and secondary metabolites. So which are the primary metabolites? Primary metabolites are protein, carbohydrate, lipid and nucleic acid are the primary metabolites. That's it. There are only four primary metabolites are there. But what are the secondary metabolites? If you look at carefully at the words primary and secondary, means what? These secondary metabolites comes from primary metabolites. So this is not topic of our discussion. We will discuss about it later wherever we will face this topic. Just today, just remember that we have primary metabolites in which we have four molecules. In that, we are going to discuss about the genetic material, that is nucleic acid. The molecular biology mainly deals with the study of this nucleic acid. Why I have told you this? Because you should get clear about why we are discussing this, what we are discussing this. We are discussing about nucleic acid, which is primary metabolite, in which you will have DNA and RNA. So many scientists worked together. In 1953, the Watson and Crick model of DNA was published. This model was very famous. In next lecture or next chapter, we have detailed discussion of the structure of DNA. There we will discuss more about the structure of DNA. But here just I am telling you the introduction of molecular biology. Up till I have told you what is actually the DNA. DNA is nothing but the primary metabolite, and nowadays it is known as the genetic material. Why I am saying that nowadays? Because this is a DNA world, means every organism have the genetic material as a DNA. But before this DNA world, there was RNA world, where RNA was the genetic material. Still, some viruses shows the genetic material as RNA inside them. But virus is not living organism. You should remember this. It's okay. We are going to discuss about the molecular biology with respect to the DNA, in which I have just told you the introduction. Now let's discuss about the scope of this molecular biology, where you can apply this knowledge of molecular biology. Scope, or we can say the application of molecular biology. The first and most important application of the molecular biology will be, the molecular biology is useful for biotechnology purpose. Why? Because in biotechnology, such as genetic engineering, we are manipulating this DNA. We can manipulate and we are manipulating this DNA. There are thousands of examples of genetically modified organisms. Today we are eating most of them. For example, seedless grapes. For example, in many animals, this genetic engineering experiment has been banned. But in plants we can conduct this genetic experiment. Why it is banned in animal? Because animal have emotions. And plants, up till we do not have any idea whether they have emotion or not, but it's okay. We are conducting the knowledge, or we can use the knowledge of molecular biology in biotechnology field, especially in genetic engineering, for production of genetically modified organisms. Okay, so it was the first scope of molecular biology. If you get an idea of I am talking about the second scope of molecular biology. Here I am telling you, if we get an idea about the structure of DNA, then you can use the fundamental knowledge of structure of DNA for studying more cellular reactions. Means what? I will talk about more it in central dogma of molecular biology, where we will see how DNA works inside the cell. Just remember here, the scope of molecular biology is useful to understand the fundamental processes which takes place inside the cell. So molecular biology does not include the study of only of DNA. It includes the study how the genetic information passes from DNA to the other molecules. So the scopes of molecular biology can be concluded in one word, is that the molecular biology knowledge is very essential in order to apply it on biotechnology purpose, because after all biotechnology is our interested area where we can create an innovative ideas, or we can have the solutions for problems which we are facing nowadays. For example, for increasing the food production, the increasing the size of fruit, such problems can be solved with the help of this biotechnology. For that, you should have the knowledge of molecular biology first. So the third and most important topic of this lecture is central dogma of molecular biology. So what is this central dogma? In one word, we can say the information can passes from the DNA to the RNA, from the RNA to the protein. Why these steps are taken place, first of all, is the most important question. Because if you look at the cell have many organelles. Out of the other organelles, nucleus is one of the most important organelle, because inside the nucleus you have your DNA. DNA is present in the form of coil and supercoil structure. This supercoil structure is consisting DNA which is wrapped around the histone protein. DNA and histone protein creates the supramolecular structure which is known as chromosome, which you know very well. We already have discussed about the chromosome. This chromosome is present inside the nucleus, in which you have your DNA. This DNA do not go outside the nucleus, because the information in this DNA is very valuable. This DNA consists such sections which are known as genes. These genes, what is gene? Gene means what? The functional portion of the DNA. What do you mean by the functional portion? Means what? I am talking about the central dogma of molecular biology. The functional portion of the DNA is going to produce the copy of the message in the form of RNA. DNA is permanent genetic material which produces temporary RNA molecule. There are three different types of RNA molecules in the cell, such as rRNA, tRNA and mRNA. The DNA produces this RNA. RNA go outside the nucleus, and outside the nucleus there is cytoplasm. In the cytoplasm, with the help of three different RNAs, mRNA, rRNA and tRNA, the information which is present in mRNA means messenger RNA, is read by the rRNA which is present in the ribosome. So the rRNA read the information on mRNA. Why it reads the information? Because the tRNA brings the amino acid for the information which is present in mRNA, and in this way all amino acids are joined together which are brought by the tRNA. In this way the protein synthesis takes place inside the cytoplasm. Means DNA is located in nucleus, it produces temporary copy. This temporary copy is mRNA which go outside the nucleus in cytoplasm. In cytoplasm, this mRNA is read and produces protein inside the cytoplasm. So DNA to RNA, RNA to protein information has been flow. You can say it is the central dogma of molecular biology. So it is very essential for biology student to understand the central dogma of molecular biology, because you cannot skip this point. As a biology student, it is your responsibility to understand each and every aspect of biology, not for the exam point of view, but as a biology student you should know about the central dogma. Just I have said you that information from DNA is passes to the RNA, and from RNA it passes to the protein. In protein you will have the chain of amino acid. These amino acids are connected to each other by peptide bond. But which amino acid should selected first? Which amino acid should selected at second number? Which amino acid should selected for the third number? is decided by the mRNA sequence. And from where this mRNA came, it came from the nucleus, from the fragment of the gene. So in case of human being, we have 35,000 genes working. Up till we have understood this, in Human Genome Project, we have found out that out of 100% DNA, please listen carefully, out of 100% DNA, 1.8% DNA is functional. So 98.2% DNA, which function is that DNA is doing, we don't know. So the functional DNA is known as euchromatin region and non-functional DNA is known as heterochromatin region. So that functional region, which is 1.8% only, is euchromatin, is going to produce all the proteins inside the cell which are very necessary. The human being which is present in front of you is made up of these 35,000 genes. So all genes are not expressed in one cell. Please try to understand this. Gene expresses in two conditions. In first condition, it expresses with respect to time. In second condition, it expresses with respect to space. Means genes which are required for mustache will express in this region only. Genes which require for eyebrows will express in this region only. Genes which are required for eyes will express in this region only. Genes which require for finger will express in this region only. So every cell contain all genes, but all genes do not express in single cell. They express where that cell is located. So this is more complicated. In MSc you will have more information regarding this. So today we just have discussed about the introductory point of molecular biology. In second lecture we will discuss about the structure of the DNA, which will be very interesting. In exam point of view, many questions can be designed on that topic. This chapter is introductory and you should understand what is the central dogma. That will be sufficient. So if you like this lecture, please subscribe the channel and don't forget to click on the like button. So it will encourage us. Thank you for your time. We will meet in the next lecture. Bye.